And hi, Nano people. My name is Olivier Fedrigo, and from the Rockefeller University, and today I'm going to present about the Vertebrate Genomes project. So this presentation will be split in two parts. The first part, I will do the first part, where I will show what the project is about, our first results and challenges. And in the second part, Arang is going to present about more in depth what the assembly process is and what are the challenges with assemblies. So the Vertebrate Genomes Project, VGP, is a project of the G10K consortium. It aims to generate near error-free reference genomes for about all the species of vertebrates, about 71,000 species. So this is a large consortium. We have about more than 150 members across 50 institutions and 12 countries. There are 3 data hubs, one at Rockefeller University, in my lab, and one at Max Planck Institute, Dresden, and Wellcome Trust Sanger Institute in England. And this is a group from my lab, and Eric Jarvis is the chair of the G10K consortium. So 71,000 genomes, that's a huge endeavor. And to be honest, right now the technology we have is not feasible, right? Maybe in 10 years, that's going to be the ultimate goal. So we split the project in 4 phases. The first phase, we selected one species per order, one representative per order, based on classical taxonomy, but also divergence time. Try to have 50 million years divergence. So we're in the middle of phase 1 right now. Phase 2 is one species per family. That's about 1,000 species, and we plan to start it around 2021. Phase 3 will be one per genus, 10,000 species, and ultimately all the species, 71,000. So these are the beasts on our list for the 261 species we're working on right now. So we have about 58 mammals, including 9 marsupials and 1 monotreme. We have 52 birds on our list, 32 reptiles, 21 amphibians, and 90 fish, including cartilaginous fishes and jawless vertebrates. So we aim to do near perfect genomes, which is hard to achieve, but that's what we aim for. A genome, of course, you all know that, is defined by their contiguity, completeness, accuracy in terms of assembly accuracy, but also base accuracy. So the VGP set up standard to say this is a VGP type genome, and that's what we want to do for phase 1, this level of quality. So instead of calling chromosome level genomes, we try to put a number behind this. So what we're looking for is NG50 contig, at least 1 megabase, N50 scaffold, at least 10 megabase. So at the time we set up this minimum matrix, we thought it was very, very challenging, but you will see that this are easily reached, except for a few cases. And 90% of the genome is assigned to chromosomes. So that's the completeness I was talking about. QV40, and some sort of phasing. Real phasing is not possible unless you have a trio, but we try to do some phasing. So before diving into this huge project and select all these species and start to sequence, we wanted to figure out what were the best technology to use and what combinations of this technology we need to do, what order of them we need to do. So about 3 years ago, in collaboration with Arang, Adam, and Sergey, who is here, we took a tiny bird, which we never understood why we took a tiny hummingbird for this benchmarking. We ran out of tissue very fast, but we tested all these technologies. So we tested Illumina short repairs. We had some Sanger sequences packed by a long ways Oxford nanopore. And for scaffolding, we tested these different strategies, including NRGene, 10x Genomics, BioNano, Chicago Laboratories, Dovetail, and a bunch of different types of IC. And we tried all the various combinations and tried to decide what technology is the best to use, not only to reach a minimum standard matrix, but also in terms of cost. So we ended up to do a kitchen sink approach where we selected only 4 technologies. Of course, we use long reads to the contigs. But then we added optical maps for scaffolding, link reads for scaffolding, and Hi-C for scaffolding. So 3 technologies for scaffolding and 1 for contigs. And for long reads, we chose PacBio, optical maps BioNano, linked with 10x, and Hi-C we chose ARIMA. So this is for phase 1. We're in the process of reevaluating all these things for phase 2. So that's how the pipeline looks like. We currently at 1.6 version of the pipeline. Essentially what you do, you take PacBio and you assemble the contigs with a software named Falcon and Zip that allows you to split the haplotype or pseudo haplotypes. Then we take the primary contigs and we use what we call purge dubs that allows us to eliminate these fast duplicates. And Aaron is going to talk more about it. Fast duplicates can appear because the haplotype is being retained in the primary contig. So it's not really another gene. Then after we scaffold with 10x first, then we scaffold with BioNano CMAPs, Hi-C, and then 3 rounds of polishing, one with PacBio data, allows us to do some gap filling, and 2 rounds of polishing with the 10x data we take again. Once we have the draft genome, we send it to another team in St. Institute, or they do the curation. Essentially, it's a very, very crucial step in our process because it's a very manual step to correct the assembly. Look at the different supports for different scaffolds and correct it manually. Of course, all these genomes, we could do that all locally, assemble the genome locally, one person do all the assemblies one by one. That would be very a huge challenge. So we're working with GNNXS, Amazon Web Services, where all this pipeline is in the cloud. So we have a group that train students or postdocs or even PIs to do the assemblies for us. And each step they have some QC to see if the assembly works properly. So that works very well. We can ramp up very fast to do all these assemblies in almost automated fashion, but still some people to train. All right. So, okay, you can get a great assembly with great data, but to get great data, you need great DNA. To get great DNA, you need great sample. The challenge of getting great samples that are good for ultra-high molecular weight DNA is exacerbated by field collection. So whether you are in the jungle in Vietnam for 2 weeks collecting snakes, or on a boat collecting leatherback turtle blood, having liquid nitrogen on you is not feasible, right? Having dry ice is not feasible. And then after the next challenge is to be able, okay, you have collected that, I need to preserve it, I need to ship it, I need to archive it. So all these steps really are important for the stability of the DNA. So the VGP was very interested in exploring this idea about what kind of guidelines can we provide to our collaborators to collect the samples, because once the sample is collected, well, that's all you got. So we wanted to give upfront guidelines. So we created this group called the Sample Prep Group at VGP. This is all the people involved in this group, where we decided to benchmark different preservation methods and to be able to create these guidelines. So we selected 1 mammal, 1 bird, 1 amphibian, 1 fish, and 2 reptiles. And we collected the tissues. We tried different preservation methods, ethanol, DMSO, DNA guard, all guard, RNA but also we tried different conditions. Flash frozen with nothing, flash frozen with some of these preservatives, or 1 week at 4 degrees in the fridge, or 1 week at room temperature. So we tried all these combinations. And then we extract the DNA. So for this purpose, we use the BioNano Prep Animal Tissue DNA Isolation Kit, because at the time, that was about 2 years ago, 1 year and a half ago, that's the only one available. It's very intensive work with all these experiments. But we're getting there, we're almost done. We have just 1 reptile, one amphibian we're finishing right now, a frog. So we're excited, very close to finish this. And then we look at the yield first. We need enough DNA to do any work. But also we look at the quality, of course. So we use pulse-filled gels. That's the gold standard. But as you can see, it's very qualitative. To do such benchmarking with something more quantitative, so we use the femto trace, which doesn't go very far in size, but we're able to look at what is below 50 kb and quantify this as an index of degradation. So this is an example of the study that we took a reptile, and this is an example for the blood. Flash frozen, of course, is a gold standard of the blood for the blood, and you can see you have a lot of fragments around 400 kb and some very high here. So this kind of sample that, with experience, we learned that this is good to go on BioNano, for instance. But ethanol with 1 week room temperature works great. So you can take the blood from a reptile, put in ethanol. It's about 10% of the blood and 90% ethanol. And you leave it on the bench. And 1 week later, you can still get this awesome DNA. So we're very excited because it's a very good solution for the field. This being said, the lab bench is not really field condition, but we're getting there. The MSL works okay, fine. But field collection is not the only challenge. The other challenge is, well, no, it's not the challenge. Museums have huge collection of samples. And it would be a little bit sad to not exploit this source of amazing samples they have. Unfortunately, these samples have been collected 20 years ago, or collected like in rough condition, or it's a roadkill or anything. Yeah, no, that's true. They have roadkills. So we decided to test it. So we collaborated with Smithsonian Natural History Museum and the American Museum, and they sent us samples. And about 30% of them are good enough. That was very surprising. And this is 3 cases where this one was collected in 2000, this one in 1989. We don't even know what type of tissue. We think it's muscle, and we don't know how it was preserved. It's probably not flash frozen, and that works fantastically. So we don't have any predictor for that. So, but we're going to keep trying. First, museum collections, because the sample is vouchered. Specialists really identify the sample, so it's very good sample in terms of metadata, but they don't tell you how you collected. So, but it's very encouraging for us. Okay, so as I said, we're almost done with this tiny project. Don't tell my people in my lab that I call that a tiny project. with this side project. So our recommendation so far for birds is blood. Blood, because blood is nucleated. So, and that's funny because the first time we used blood, because of USDA restrictions, we had to have blood and ethanol, because they didn't want to import blood from Europe with no ethanol because of the bird flu or stuff like that. So we're like, okay, it's not going to work. And we work with BioNano on that. I think BioNano got the first blood in ethanol, and we work with, I don't remember her name now, Saki from BioNano, and she figured out a way to use blood in ethanol. And it's the best solution for us. So, and one of the reasons is because bird blood is nucleated. It's not like mammal blood. There is a lot of DNA in that. Reptiles, the same thing. Reptiles, nucleated blood, and this is very good. We like also all blood, but also isolated red blood cells. Works very well too. For mammals, blood works okay, but flash frozen muscle or spleen is really the way to go. So we try to set up all these recommendations for our collaborators, and so far we're happy with the results. So we started this project, the VGP, about 3 years ago, once we did the benchmarking of data types. And this is where we're at right now. So far, we have 121 genomes with complete data sets, and assembly completed or in progress, right? So I think about 75 of them is completed assembly, and the rest is we're still working on that, the last steps of the assembly. So we sequence 26 mammals, 35 birds, 6 reptiles, 6 amphibians, 42 bony fishes, 5 cartilaginous fishes, including a great white, and a jawless vertebrate. So that's really a team effort. As I mentioned earlier, there was 3 hubs, Max Planck, Sanger, and us. and a group NIH that helped a lot, Arang and Sergey, to do the assembly. So it's a really huge team effort of this consortium. We're very proud of this. So all this data, as soon as we generate it, is uploaded to AWS bucket. And we created this, this interface website that you can get the raw data right away. So automatically publicly available. Assembly, as soon as they're ready, your draft assemblies go there. Created assembly goes there. So if you're interested, you can look at all these genomes on our website. It's Genome Arc. I don't remember the URL. Okay, good. Yeah, she may repeat what I said before, but that's okay. So I played with that a little bit last night. I look at all the genomes that have been created. And on the x-axis, the contingent NG50, and the y-axis scaffold NG50. And you can, and you have a 10 megabase scaffold and 1 megabase scaffold threshold, and you can see most of the genomes are up there, except for a handful of fishes. Fishes are hard. But what strikes me is that you see these clusters of animal groups, and it's probably due to genome size as well. And G50 normalized for the genome size, but the chromosome length may influence N50 as well, right? So you can see mammals are usually higher N50.
Why do you think only fishes are so much represented?
Repeats, right? You're going to talk about it. Repeats. Yeah. I mean, first of all, it's a smaller genome, so you're going to plateau your N50, right? But also repeats is just tough. And getting good DNA also is very tough. So most of the time, they're shorter reads.
Which one is it? 300 megabase? That one works.
That's a frog. Oh, that's an amphibian.
No, the fish that's.
Oh. Is that the? I don't remember. Yeah, I would double check. But yeah, that's a fish, I'm sure. Yeah.
Are you fine also with those small chromosomes from birds?
The macro chromosomes, that we find some.
I mean, we go down to chromosomes. We could identify as chromosomes are based on high C maps, majority of them. We found 32, 34 chromosomes for the hummingbird. See similar results for other birds.
So
It's like going down to 28.
So here an example, I pick up 16 other birds, and this is to illustrate what the contribution of each technology. So you see PacBio reach a certain N50, you add 10x for scaffolding. So this is contingent 50, and it's going to be scaffolding 50. You add 10x, and you have a slight improvement, very, very minor improvement. You add binary, it's probably the largest jump, and after IC, you get another jump. So very consistent with expectations. Interestingly, you see some of them that they kind of plateau very quickly. That even adding binano help a little bit, and I see almost nothing. And I don't know yet why, but I put the chromosome numbers on those. And of course, the one with the larger than 50 have 27 chromosome, less chromosome, the one with the lowest than 50, 52 chromosome. And all these birds have about the same genome size, 1 to 1.2 gigs. So that means that you have large chromosome or small chromosome. So that's why N50, you have this kind of distribution like that, I think. But the question is, okay, what is the contribution of each scaffolding step? So this is a quick plot of the same 16 birds. So in blue is a PacBio with just 10x scaffolding. In green is PacBio with just binary scaffolding. And in yellow is PacBio with just IC. Surprisingly, many times you see binano being outperforming IC, and I'm not quite sure why. I may have made a mistake when I calculated all these steps, but I was very surprised to see like these large green bars and a small yellow bar. Yeah. But also, what is the order? How do you combine them? Do you do PacBio, IC, and binano, or PacBio, binano, and IC? So that's something in Hummingbird we decided to go binano first, but we want to test that with the genomes we got, right? So in yellow is really the combination of all three in the same order we did the benchmarking. But you can see that every time in green, when you add bioanalyzer first, you get better results than if you add IC first. And I was talking to Sergey about that a few minutes ago, and it's a mix. It could be the data type that binary is more accurate, but also it could be a software issue, right? A software difference. Like if the solve, the scaffolder, doesn't find any labels in this region, no support for a particular scaffold determined by IC will break it, right? So it's a combination of both, I think, data type and software approaches. So before yielding the floor to Arang, I would like to point out one of the projects that I got most excited about. It's the Vaquita Project. Vaquita is a small porpoise from northern Mexican Gulf of California in Mexico. It's a very cute little, I don't know if I can call it dolphin, it's a porpoise. A very cute little animal, 4 to 5 feet long, under 20 pounds. Fortunately, there is about 12 to 20 individuals left in this population. So 1 year ago-ish, NOAA sent an expedition to the Gulf there to find this population. It's very hard to find as well. They're very shy. So they have like this, they collaborated with the US Marines, US Navy, with dolphins. Dolphins were tracking them. It was a pretty cool expedition. They found a few individuals and they tried to capture them to put in this breeding facility. Unfortunately, these animals are very fragile and the one they captured died. So one less, that's a huge, that was very unfortunate. So the reason why right now they're low population is because they're being overfished in Mexico. Not on purpose, but it's fishing the totoba fish is very important for the community there because they can sell the bladder to the Chinese market, to China, because, I don't know. And these tiny porpoises get caught in nets. So so they try to change the regulation, but it's a big battle. So in collaboration with Olivier Ryder from San Diego Zoo and Phil Morin from NOAA, who actually led the project, we are able to culture the cells. So we have now cell lines for this animal. So one day if we do Jurassic Park, we can put it back in place. So we extracted the DNA from these cells, and for the same DNA batch, we use PacBio, Sapphire, and Chromium on this for the exact same DNA. So one extraction. Then we do that IC2. And I think the BioNano was done at BioNano. And this is the results. We got a genome of 2.3 gigs genome long, and we got NG50 of 16 megabase contig, which is pretty good. We're very excited about that. And a scaffold N50 of 100 megabase. So Phil Morin, the one on top right, did some analysis where you look at the historical demography of the Vaquita. He did a pairwise sequencing Markovian coalition model, PSMSC, where he could estimate the effective population size in time. And you can see this is today. You can go back in time, and the population has always been small. Now, that doesn't excuse what's going on right now, but that gives us hope. That means that this population could handle low diversity, right? It used to bottleneck, so very resilient. So there's still maybe some hope that we can reestablish the population. And the reason why you have this low population is because of the sea level. Since the northern Gulf of California, you see the sea level goes up and down, and they get stuck in a small corner. So the population always was small. So that's very interesting aspect of this. Right? So I don't have any acknowledgements because Arang has a bunch of them at the end. And I'll leave the floor to Arang. Thank you.